Hello everyone, my name is Alaa Sayed. I'm a medical student at King Saud bin Abdulaziz University for Health Sciences, and today I'm going to talk to you about the development of the central nervous system. This is a brief outline to the things I'm going to talk about during the video. Before we start with embryology, let's just start with the basics. Now, the nervous system is divided into central nervous system and the peripheral nervous system. The central nervous system is divided into the brain and the spinal cord, while the peripheral nervous system is basically everything else besides the brain and the spinal cord. The brain. Now, the brain is divided into the cerebrum, which is made up of lobes, the temporal, parietal, occipital, frontal lobe, and also the diencephalon, which is basically the thalamus and its family, so thalamus, hypothalamus, subthalamus, epithalamus, and then the brainstem, which is made up of the midbrain, the pons, and the medulla oblongata, and lastly, the cerebrum. This is a picture of what I just talked about. So the cerebrum here, the diencephalon here, and then you have the brainstem, which is the midbrain, pons, and the medulla oblongata. Now, the continuation of the medulla oblongata downwards here is what we call the spinal cord. Now, let's start with some basic embryology. Now, this is the ovary, and the ovary will release an egg. Now, the egg will get fertilized by the sperm, and then it will make up the zygote. Now, the zygote will start replicating or dividing into smaller and smaller cells, and then it will eventually give the embryo. Now, as you can see here, it's moving through the fallopian tubes, reaching the uterus, and it's dividing while doing that until it reaches a stage called the blastocyst. So the blastocyst is the stage that implants within the uterus. Now, this is basically the same thing. After the formation of the zygote, after fertilization, you will get the embryo, which will start to divide, okay? It will give two cell stage, then four cell stage, then it will give something called a morula. As you can see here, there's a membrane surrounding that embryo, and this membrane, it like limits the embryo from getting bigger. So as you can see, the cells are dividing more and more, but they're smaller in size. So basically, the morula is just a ball of cells which are compacted next to each other. Now, when this ball of cells start developing a cavity, as you can see here, we call this stage a blastocyst. Now, the blastocyst is basically the same thing as a morula, but it started developing this blastocyst cavity. Well, but that's not the only change in the blastocyst. Now, in this phase, as you can see here, we have different colors of cells. You have the blue ones and the green ones. Now, the blue cells, or the cells in the middle, are called the inner cell mass, or the embryoblast. Now, those cells will actually give you the embryo. That's why they're called the embryoblast. While the cells surrounding them, or the green ones, are called the outer cell mass, or the trophoblast, and they will eventually give you the placenta. Now, we won't focus on the outer cell mass. We're not interested in the placenta right now. We'll only focus on the inner cell mass, or the embryoblast. So the whole embryo comes from the inner cell mass. So whatever differentiation is going to happen in the embryo, it's going to happen in the inner cell mass. The bilaminar disc. Now, what happens here? Remember the blastocyst in the last picture? Now, the inner cell mass started to differentiate into two layers of cells. One called the epiblast, which is blue in color, and one called the hypoblast. Now, those two epithelial layers start developing cavities within them. So within the epiblast, you have the amniotic cavity, and within the hypoblast, you have the yolk sac. And this is what we call the bilaminar disc, because it's two-layered phase of the embryo. One is the epiblast, and the second is the hypoblast. Now, this is another picture of that bilaminar disc. Now, I need you to imagine something. This disc is not as we see it right here. Now, basically, this is just a cross-section. This is a cut. Imagine the embryo is like this, and this is where you're taking your cut. So those layers, like the epiblast and the hypoblast, are like here. They're only one cut of the system. Now let's just imagine we're taking them as a disc, like a real disc. So this would be your epiblast, and a layer down below it would be the hypoblast. Now what happens to this disc? Just imagine with me. Now this disc is full with cells of the epiblast, right? Within this layer of epiblast, or this plate, you have two membranes, okay? One which is here, and the other on the other side. Now this is the cranial end, and this is the caudal end, okay? Now on the cranial end, you have the oropharyngeal membrane, which will give the future mouth of the baby. And on the caudal end, you have the cloacal membrane, which will give the anus of the baby. Okay, so imagine this was our plate. Okay, now what happens in this picture? As you can see here, we have the epiblast, the hypoblast. So basically first what happens is that some cells of the epiblast will start to degenerate or die. Those cells will give us what we call the primitive streak, which is just a line of empty cells that died or degenerated. Now, and then after a while, that primitive streak will even develop what we call the primitive node. So more and more cells will degenerate, as we can see here. This is a primitive streak, and then the primitive node at the end of the primitive streak, which is just a bunch of cells that degenerated. And then what happens after that? Not only did the cells die, some cells from the epiblast, from here and here and here and here, will start to migrate from this opening, or from the streak and the node. Now, cells migrating from the streak will give a different structure from cells migrating from the node, and we'll take them one at a time in the next slide. So, let's start off with the cells migrating from the primitive streak. Okay, this is a primitive streak and this is a primitive node. Now the cells migrating from the primitive streak will start to invaginate downwards towards the hypoblast. And as they go downward, they start to make the layers of the embryo, the other layers of the embryo. So they will give you the mesoderm. This is the mesoderm. And then they will go towards the hypoblast and start giving you the endoderm. And the cells that will stay here will transform into the ectoderm. So basically what happened here? All the cells going through the primitive streak will give you the embryonic like layers, the endo and the ecto and the mesoderm. And this is what we call the trilaminar disc, or the three-layer disc, okay? And now let's go to the cells coming through the primitive node. Now the cells going through the primitive node, as we can see here, this is a primitive streak and this is a primitive node. Well, actually, as you can imagine, they will go downwards. So they're going downwards toward what? Toward which layer? The mesoderm. Imagine cells going through the node, they're going through the mesoderm, okay? So first of all, they're going through the mesoderm. And the second point is they don't start from the beginning. As you can see, the node does not start like the streak from the beginning. It starts at the end of the streak. So they will start at the middle, at the mesoderm, and move cranially towards the oropharyngeal membrane, or towards the future mouth. And they will make a structure, as you can see here, that it's like a line or a tube. This structure is what we call the notochord. You can also see the notochord here in this picture. This is the notochord. This is another picture of what we just talked about. So as you can see here, this is the ectoderm, and here you can see the notochord forming from the node going downwards from the primitive node. And yeah, this is a notochord. It began from the middle, and it's also, as you can see, moving cranially toward the oropharyngeal membrane, and also it's in the middle layer, or the mesoderm. Now what happens after that? This is another picture. See the notochord within the mesoderm of the embryo. You can imagine what the notochord starts to do. The notochord starts secreting growth factors. Those growth factors will act on the ectoderm above it. Now the ectoderm above it will start differentiating into neuroectoderm cells, or will start like multiplying in the middle, here and here and here and here, into neuroectoderm cells. This is the same thing we talked about. So this is the notochord. The notochord is secreting growth factors. The ectoderm above is differentiating into neuroectoderm cells, getting bigger and bigger, bigger and bigger, until they give us something called the neural plate. This is the neural plate or the neuroectoderm cells. Now, it's basically a bunch of ectodermal cells above the layer of the mesoderm. Now, what happens to this neural plate? Now, as you can imagine, the streak is still there. So those cells from the neural plate will start to migrate through the streak downwards towards the mesoderm. As you can see here, let's imagine our neural plate as a book. Okay, so let's imagine it. I'm bad at drawing, but bear with me. This is my book. It's over a table or something. So this is the book. We have the notochord below. Okay, so our neural plate, as you can imagine, if we have a streak here, the book will start to close on itself, right? So as you can see, when the book starts to close, it will give you what we call the neural groove, because it's grooved downwards. Okay, so this is a neural plate. It starts to groove downwards from the streak, giving us the neural groove. And now some cells at the end of the book will just stay stuck in the ectoderm. And this is what we call the neural folds. Okay? And then when the groove goes downwards even more, it actually joins each other, the sides joins, and they close, giving us the neural tube. And the cells that were stuck as neural folds upwards will go downwards, and they're like, okay, he's going to the mesoderm, I'm going too. But they get cut off separately as neural Crest cells. This is basically a repetition of what we just talked about. So let's just sum up. Okay, you have the notochord. Notochord starts stimulating the ectoderm. The ectoderm forms the neural plate. Neural plate starts migrating toward the primitive streak downwards. When it migrates, it gives us a groove. The edges of the groove are called the neural folds, which are still in the ectoderm. When the groove goes down, it joins each other, it closes as the tube, and the neural folds get cut off and goes downwards towards the mesoderm as neural Crest cells. Now, the important thing to mention here is that this neural tube is not empty. It's filled with a CSF fold cavity. So as you can imagine, as this neural tube is developing into further brain structures, every part of the neural tube will also give a cavity with it, a CSF fold cavity, or it will give, in other words, a ventricle. Now, this is another picture of the neural tube. So the neural tube has two endings, a cranial end called the cranial neuropore and a caudal end called the caudal neuropore. Now, the cranial neuropore and the caudal neuropore both close normally in the embryo. So the cranial neuropore closes first at day 25, and then 3 days later, or at average at day 27, the caudal neuropore closes. Failure of closure of those two neuropores will give you congenital anomalies, okay? So let's imagine we had our neural tube. Now what happens to this neural tube? First, this neural tube starts developing 3 swellings, okay? Those swellings are called the 3 primary vesicles. As you can see here, this tube developed 1, 2, and 3 swellings. The first one is called the prosencephalon, or the forebrain. The second one is called the midbrain, or the mesencephalon. And the third one is called the rhombencephalon, or the hindbrain. So basically, you need to memorize those 3 names, the prosencephalon, mesencephalon, and rhombencephalon, before you move on. Now, what happens to those 3 vesicles? Now, let's take the prosencephalon. Now, the prosencephalon has 2 parts. This part and the lower part and the upper part. Now the lower part actually grows relatively slow, so it will just elongate a bit, as we can see in the next picture. And the upper part will actually grow more faster, but it will grow a bit funny. Now it will go faster at the edges, laterally, so at the right and the left, and leaving a bit of a constriction in the middle, growing a bit relatively more slower. So it will give you a shape like this because it's growing really fast, and then a constriction in the middle, and it's growing really fast in the other edge. So it will give you actually Mickey Mouse, okay? A structure like Mickey Mouse. Now, those two structures will be called the telencephalon in the upper part. So imagine telescopes are always in the upper part. And then the structure that was downwards will be called the diencephalon. So people who die are buried down in the ground. So this is what I basically just talked about. So let's take the first part, or the prosencephalon. Now the prosencephalon, as you can see here, this part, the lower part, which is the diencephalon, will go relatively slow. And the diencephalon will eventually give you the thalamus and its family. Okay, and then the upper part, which is the telencephalon, or the telescopes, as you can remember, Mickey Mouse. Now, Mickey Mouse will give you the cerebral hemispheres, so the parietal lobe, temporal, occipital, whatever. Okay, and this is for the first swelling, or the prosencephalon. Now, the second swelling, or the mesencephalon, will actually stay the same. It's in the middle. It doesn't like change, so it just stayed the same. Okay, now the rhombencephalon says, okay, mesencephalon stays the same. I'm changing into two things. So it gives us two other swellings. This is the first swelling, and this is the first swelling, and this is the second swelling. Now the first swelling is called the metencephalon, and the third swelling is called the myelencephalon. Now, as you can see here, the metencephalon will eventually give you the pons and the cerebellum, and the myelencephalon will give you the medulla oblongata. Okay, why did I put this picture here? I need you to imagine how the brain looks right now to know how did it come from. Now, as you can see, this is the telencephalon giving you the cerebrum. Okay, and then in the middle, or a bit downwards to the telencephalon, you have the diencephalon, okay, giving you the thalamus and the thalamus family. Now, then downwards, you have the mesencephalon, giving you the midbrain. And then downwards, you have the rhombencephalon. Now, the rhombencephalon, as we mentioned, it has two parts, the metencephalon and the myelencephalon. So, we mentioned the metencephalon will give you the pons and the cerebellum. So now this makes sense because they come immediately after the mesencephalon, like the metencephalon. The metencephalon comes immediately here. And the second thing to mention is, can you see that the pons and the cerebellum are actually at the same level? So it just makes sense that they come from the same structure, or they both come from the metencephalon. And then, under the pons, as you can see, you have the medulla oblongata. So the medulla oblongata comes from the structure that comes under the structure that gives the pons. So if the pons came from the metencephalon, the medulla oblongata will just come from the myelencephalon below. This is a repetition of the previous slide. I just need you to focus on something. Here we're going to talk about the origin of the ventricles. Remember, we said the neural tube is the CSF-filled cavity, and as it grows, it will give you the cavities or the ventricles with it. Now, just for the cavities or the ventricles, imagine their position now in the normal human being, like an adult human being, or a born baby, and just reverse the origin back, and you'll know where did they come from. So, as you can imagine, the lateral ventricles are actually situated within the cerebral hemispheres. So they come from the same origin as the cerebral hemispheres, from the telencephalon. The third ventricles are found within, like between the thalamus, the two thalami. So they come from the same origin as the thalamus, so from the diencephalon. The aqueduct of Sylvius is situated within the midbrain, so it comes from the structure giving the midbrain, that is the mesencephalon. The fourth ventricle, however, actually is found within the pons and the medulla. It comes from the same structure that gave the medulla and that gave the pons, which is the metencephalon, myelencephalon, eventually coming from the rhombencephalon. So this is just for you to imagine. So this is the lateral ventricle. It's within the cerebral hemispheres here. This is the third ventricle within the thalami. And this is aqueduct of Sylvius within the midbrain here, and the fourth ventricle within the pons and the medulla oblongata. Now, the development of the spinal cord. I just need you to know 4 terminologies here. So basically, we first started off with the neural tube. We mentioned the neural tube. How does it give the brain? Now, how does the neural tube give us the spinal cord? Basically, the neural tube is lined with epithelial cells. Those epithelial cells are what we call the neuroepithelial layer here in the middle, as you can see. They're also called neuroblasts. Now, what are the neuroblasts? The neuroblasts are cells capable of dividing more and more, giving us more cells outwards or more neurons. So as they divide, they will give you cells here on the edges, okay? And this layer of cells or neurons are what we call the mantle layer. So the mantle layer are new neurons, or they are cell bodies in specific of those neurons. So this is what we call the mantle layer. As you can imagine, if they are cell bodies, then they make up the gray matter of the spinal cord. And then those mantle layers will start actually giving off axons or receiving axons, depending on whether they're sensory or motor. So as you can see, let's take the dorsal part. Now, the dorsal part is actually sensory. So the sensory part or the dorsal part will actually receive axons from outside, okay? And the motor part or the part in front will actually give off motor axons. Now, the axons going out from those cells here and here give us something called the marginal layer. So what is the marginal layer? The marginal layer is axons of the cells. They get myelinated and they form the white matter of the spinal cord. So the mantle layer, cell bodies, gray matter. The marginal layer, the axons of those cells get myelinated and they form the white matter. Now, what is the alar plate or the alar plate? I don't know the pronunciation. And what is the basal plate? It's basically parts of the mantle zone divided according whether they are sensory or motor. So anything called alar or alar is sensory, and anything called basal is just motor. Okay, so derivatives of the neural crest cells. As you can imagine, those other cells we said got cut off, so they surround the neural tube. Remember, I mentioned the neural tube will develop into the spinal cord. And then you have this part will be sensory, which is the dorsal, and this is motor. So the neural crest cells are more towards the sensory part. Okay, and remember they are ectodermal in origin, as we mentioned, and then they will migrate laterally and give rise to the sensory ganglia. And this makes sense because they're more towards this part, and this part will actually develop later into the spinal cord and then the sensory component of the spinal cord. And then, as you can imagine them, they're near this part of the cells, so they're near axons. So they'll eventually give you the sympathetic neuroblasts, the Schwann cells, pigment cells. They also give odontoplast and the meninges and mesenchyme of the pharyngeal arches. In this figure, I just needed to summarize everything, so you go through it. The additional point here is that you have 12 cranial nerves. Now, where do they actually originate from? It's easy. Let's just memorize it. You have 12. All of them originate from the hindbrain except for 4. 2 of them will originate from the forebrain and the other 2 from the midbrain. We mentioned the forebrain had 2 parts, so it just makes sense that one nerve, which is the first nerve, the olfactory, will come from the first part of the forebrain or from the telencephalon, and the second nerve will come from the second part of the forebrain, which is the diencephalon. And the third and fourth nerve will come from the midbrain, and the rest will come from the hindbrain. Okay, these are my references. I'm done. If you have any questions, any comments, just contact this email.